Surbhi, do you wanna? Surbhi's question is, what is the difference between RNA and DNA sequencing?
So RNA is the transcript. It's what is transcribed from the DNA. In the DNA, it is telling you which genes do you have. The RNA is actually expression of that gene. So you can so in terms of the methodology, when you're looking at the sequence, it looks exactly the same. There's no difference, but it's just what part of the body you're measuring. So it may happen that you know that your DNA is coding for, let's say, FLT3 genes on chromosome 13. But and if you just looked at the DNA, you're like, yeah, okay, I have an FLT3 gene, I know the sequence is there, and you will just assume that it is also expressed in your body. But if you did RNA sequencing, you might realize that, oh, my FLT3 gene is actually underexpressed or it's not expressed at all. So in RNA sequencing, one important step that we don't really care about in DNA sequencing is number of copies of the RNA. So not every patient will have the same amount of FLT3 gene expressed. And so maybe in you it's 10 times, maybe in me it's 15 times, maybe in Dr. Watman it was probably like 50 times. And so that's the major difference when we are trying to interpret these two sequencing.
Aditya has one question. He's asking, why does radiation affect our DNA?
Yeah, so radiation affects our DNA because it breaks down a very complicated machinery around the DNA that keeps it working the way it should. So there's something called DNA damage repair. What happens is that your DNA is double stranded, right? You might have seen those textbook images of a helix of DNA. And your DNA is continuously being read and synthesized. So how does RNA get transcribed from DNA? DNA is like a helix, right? For a while, it will open up. The some complicated machinery or molecules will come read the DNA and make your RNA, and then it would go back and it would close up. For this entire process to keep working perfectly every single time, there are other supporting molecules that keep our DNA in check. If there's any place where, say, DNA gets damaged, has a break, is not binding back correctly, those proteins and machinery comes into place and fixes it. But radiation or chemotherapy actually impairs that machinery. It breaks down the molecules and the proteins that fixes the DNA. So a lot of the cancers that we study today are actually driven because of a mutation. Um, some of them may be driven because of radiation. So you guys might have heard of the nuclear accident in Chernobyl, right? Um, or the nuclear bomb that was dropped in Hiroshima and Nagasaki. Just yesterday, a study came out that studied the impact of radiation on, I want to say, around 1,000 and more individuals, and studied how mutations caused by those radiations has led to cancer of different areas in the body because of those radiations. That is a recent study. Another study that is not radiation associated, but we know that cancer is caused by that mutation, is breast cancer. Breast cancer, or almost like 1 in every 3 women in the US who have breast cancer, of patients who have breast cancer, 30% of them have a mutation in their gene called BRCA1 or BRCA2. It's only because of that mutation that they got breast cancer. So a lot of the cancers are actually driven through mutation. Some cancers are more heavily driven by mutation. Some cancers are more environmentally driven and not really by mutation. For example, one of the cancers that I've studied, stomach cancer. Um, if you ever end up studying stomach cancer, you will, one of the things you'll study is that people in Japan and Korea and Asia have very high rate of gastric cancer, but people in the US, not really. Now that number is kind of equalizing. But if you look at historical trends, Japan and Korea used to have a lot of cases of stomach cancer. Then I tried to find out, okay, why is that happening? One of the surprising reasons that came out is because people in Japan and Korea at the time did not have refrigerators, but people in America did. So, and these guys used to have fermented food that was kept outside for a very long time. And when they used to eat that, that food actually had a bacterial infection, but they didn't realize it. They didn't have refrigerator. That was just their lifestyle. When they ate that food continuously over a period of time, the bacteria in their stomach, the levels of that bacteria in their stomach obviously went beyond normal or healthy levels, and that bacteria ended up giving them stomach cancer. So just to give you an example of how this DNA editing works, in case you guys are already aware, just let me know. I won't teach you what's in your books. Um, but the whole idea is that we want to fix these changes in the DNA that are causing us problems. So Jennifer Doudna and others, they identified from a bacteria an enzyme that can actually cut DNA. And that is what led to this whole CRISPR-Cas9 revolution. That if we actually somehow direct that DNA cutting enzyme just to that right place, just to that specific base pair, then we can get that defective copy or that mutation removed out, and automatically the body will start producing a healthy DNA strand. So how they did that is they identified, what they did is they made guide molecules, which is a mirror image of the DNA, the other strand. So if it was A, they'll make T. If it was G, they'll make C, so on and so forth. And they kind of attach that CRISPR-Cas9 enzyme onto that guide RNA. And our body is so amazing. If we insert that guide RNA into our body, it knows where to go and where to bind in the DNA. Once it binds, the DNA cut enzyme, now that it is bound and is activated, will cut the DNA exactly at that right place. And then what our assumption is that our healthy DNA polymerase machine is going to come in, make a new strand, and effectively replace it.
But
Um,
so I have a question here. So our body is constantly producing cells. Cells have nucleus. Nucleus has DNA, etc., etc. I mean, how often do you do this? How do you manage to do this across millions of cells? Like, that's
yeah.
what's a little confusing here.
Yeah, so there won't be one guide molecule that we'll provide. The guide molecule will have multiple copies that we'll inject into the cells. Our hope is that enough number of healthy DNA will lead to production of. So your the strands that have the incorrect or the defective DNA strand is not going to go away immediately. So you can't make it go away, but you're kind of overwhelming it with the right thing. So our point is to overwhelm the cell to a point where only healthy strands are being produced, and then leave it to the cell to do. But obviously, it's not 100% accuracy that from today that's it, no defective DNA strand will be produced. So the other thing that we can do with genomics, apart from cancer or genome editing, is actually creating effective vaccines, which is something you guys must have heard for COVID-19. But the other thing that happens routinely, but we don't realize, is actually flu shots. So in India, flu shots are not that common. But outside of India, in countries like Singapore or Canada or the US, flu shots are recommended every year to people. Every year around when spring comes in or late October, they're recommended that please take flu shots. So why they do this is that the labs or the government health laboratories that are tracking influenza, they keep randomly taking samples from people who have had the flu virus as well as not. So they'll take random samples and they expect a certain proportion of the population has flu virus. And then from those samples, they will actually sequence the genome of those influenza viruses. Why? Because influenza viruses like COVID vaccine, the COVID-19 that you're hearing, undergo mutations. So they want to make sure that the flu shot every year people are getting it's actually protecting them against the latest form of this influenza. There are many types of influenza viruses. So this vaccine is kind of a combination vaccine that could treat up to 50 types of these influenza viruses. Pregnant mothers today can go to the clinic, and it is a part of recommended tests in many countries where they take the amniotic fluid, and that can and they also have ways. I'm not sure exactly what path they take, but they have ways to sequence the genome of the unborn child as well, and determine if that child is at some predisposed risk of some fatal disease or Down syndrome or autism or other things, or maybe something that's being passed down from the mother um, to the child. Have you guys heard of this company called 23andMe? So basically, it's called 23andMe because you have 23 pairs of chromosomes in your body that make you you, right? And what they claim to do, and they have actually successfully shown as well, you can take a sample. You can take your saliva, and it's a kit that they sell you. Okay, so you can sign up, you can purchase their kit, and they will send you this box with like this picture that I've shown, like a small tube, a swab, and a few other things. What they want you to do is they want you to give you a little bit sample of your saliva, and they want you to swab your inner cheek, get some cells from there and put it in this tube and ship it off to them, right? So you what they will do is they will take those cells and those saliva, they will break that down, extract the DNA, extract the RNA, mostly DNA, and they'll sequence it. Now that they have sequenced it, they have a repository of many other sequences, right? So, like Aditya said, they will try and map it to other genomes that they know are of, let's say, Irish descent, or like some Nordic descent, or some other genomes that they have from Africa or East Asia. And then they will try and tell you, okay, you have 75% match to Southeast Asian descent, and you have 2% of some Iranian descent, and 5% of something else. Those are like fun things. Or maybe somebody really doesn't know what their family line is. Their family has migrated across regions, and they are very curious. They can get their ancestry test done, or for more meaningful things, they will check and tell you if you have the BRCA1 or BRCA2 mutation, because they know what a BRCA1 BRCA2 healthy gene sequence looks like. There are other examples as well. There is a gene called cytochrome 560, which determines your response to depression medicine. So if people who are suffering from depression and are given a certain medication, some people have very serious side effects because of mutations in that gene. So then they'll tell you, oh, if you ever have depression, I think you have this mutation, maybe you should discuss with your healthcare worker before taking that medicine. So because of this whole thing of getting your genome sequenced so cheaply and so quickly, these direct to consumer testing has happened, where you can ask the question if you're predisposed to a cancer, you can ask the question if you're predisposed to some other disease, you can get to know who your ancestors are. There was a very interesting example of a researcher in Israel whose family had migrated to Israel after the Holocaust, and obviously a lot of their family was still missing, and he didn't know a lot of his previous family, and he got his 23andMe test, or I think some other direct to consumer testing that I'm not sure if it's 23andMe, but basically he wanted to test his ancestry. And you can imagine other people like him were doing it, and he signed up for this option where they said that if we find a really close match of your DNA to some other consumer who comes to us, we will let you know. And that way he found like a second cousin of his who had long lost, and they found out, oh, we share a lot of genetic similarity, it seems like we have the same common lineage. And when they connected, they actually found that they had common relatives who were separated during Holocaust.